Okay. Thank you very much, Arnold, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, yeah, then thank you for the invitation to give a talk about our synthetic cell work. Well, Zoom meeting reminds me of a time which I do not want to be reminded too much, which is the Corona time, of course, where we did this very often. Nowadays, it's far less. And the problem is, of course, always with Zoom is that you don't have the direct contact with the audience. But okay, this is what it is, so we have to do it like this. So today what I will do is tell you a little bit about our work on synthetic cells, and in particular phospholipid biosynthesis in synthetic cells and associated processes, because we also used those developments to address some scientific questions in other systems we are working on. Now, we're part of a big Dutch consortium, which is called BASIC. The logo is indicated here, where a large group of researchers works on a common theme of a synthetic cell. And let me go to the next slide. I would like to go to the next slide. Yes, there we are. Okay, so building a synthetic cell. So what we're talking about is a bottom-up approach. So to build a living cell with a minimum number of components from scratch. So DNA, RNA, enzymes, lipids, and so forth. The ultimate goal of the synthetic cell is that it becomes self-reproducing. So there must be growth and division of a compartment. This is the artist's impression of a cut-through of a synthetic cell where we have some of the basic components indicated. But of course, it's an artistic impression. Now, of course, this is a formidable challenge because building a cell is not a simple thing to do. Well, I mean, may take some time, but while working on such a project, you can also start to learn about biology. How do things work together? So what are the challenges to construct a cell from molecular components? Well, it's manifold. So first of all, you need some kind of energy, fuel, ATP, maybe electrochemical potential and redox energy. You need the building blocks. Well, the building blocks, there are different ways. You can synthesize those building blocks in confinement, or you can transport, so feed the synthetic cell with the molecules it needs. But it should also be physicochemical homeostasis, so there should be a stable internal pH and ion composition. Well, the other part is information storage and processing. So the synthetic cell eventually needs to be encoded on an artificial DNA, and this DNA needs to be transcribed, of course, transcribed into RNA and eventually into protein translated. So protein synthesis needs to be encoded into this synthetic cell. Then we have growth and division. So much of that is based on self-organization, at least that's the assumption for building a synthetic cell. We need to grow membranes. And these cells, if they grow, they need to divide somehow. And then you get into more complex matter, communication, adaptation, and repair. And these are all elements which are addressed in this basic consortium. But what I will focus on today is membrane growth, because that's the part we are dealing with. So membrane growth in a synthetic cell. So what we did to approach this is we follow an in vitro approach. So we purify all the protein components needed for phospholipid biosynthesis, put them together into a liposome, so reconstitute into liposome, and then feed with precursors to grow these membranes, to grow them with newly synthesized phospholipids. And for this first part, I have to say this we do from the outside. So we start with liposomes, and we let those liposomes grow by giving precursors and synthesizing phospholipids. Now then you need simple components, and so typically what is shown here is the phospholipid biosynthesis pathway of E. coli, at least one of the variants. And so this normally starts with acyl-CoA as a precursor for the fatty acid, glycerol 3-phosphate, and then this peripheral membrane protein, Pils B, which converts these two into lysophosphatidyllic acid, which is then further converted by Pils C, another peripheral membrane protein with another acyl-CoA, into a phospholipid phosphatidyllic acid. Now then the pathway goes on, and the rest of the pathway involves a number of integral membrane proteins, so membrane spanners, multiple membrane spanners. A key enzyme is CDSA, which is a CTP transferase, which activates the phosphatidyl acid group with a CTP. Then you get CTP-DAG, CTP diacylglycerol, which is a precursor for polar head group attachment. And then the pathway splits up. So it can either be used to form phosphatidyl ethanolamine, which is one of the key lipids in E. coli. About 70% of its membrane is PE, unsaturated PEs. Or it can be converted into phosphatidyl glycerol in E. coli. So we follow the E. coli pathway. Now, phosphatidyl glycerol is an acidic phospholipid. It's an essential component of the membrane. The same holds for phosphatidyl ethanolamine. It's with ionic phospholipid, but it forms non-bilayer structures, and this polymorphism is also an essential feature of the membrane. So we deal with these two properties. Now, normally the acyl-CoA comes from fatty acyl synthetase, and that's a very large multi-subunit protein, very complex. And although in the meantime, people have reconstituted that also in the context of this part of this pathway, we felt we need a more simpler solution to that. And that's actually using a catabolic enzyme from fatty acid degradation called FAT-D. Now, FAT-D is involved in fatty acid metabolism. And what it does, it takes any type of fatty acid and activates it with a CoA at the expense of ATP, and then you get an acyl-CoA. So we have a simple entry of acyl-CoA into this pathway by feeding with fatty acids, CoA and ATP. Now, so the postdoc in my group who did most of this work, Marta Ekskater, he overexpressed all these proteins, purified them, and reconstitute them into liposomes, and then feed with the essential components. Let me go through some of those enzymes in a stepwise manner before I go into the growth. So first, FAT-D, it's a versatile CoA activating enzyme of all kinds of fatty acids. In this example, what we look at is oleic acid, which is we mostly use. So what we do is we have the purified enzyme, we feed it with the oleic acid and the coenzyme and ATP, and then analyze the products by LC-MS. And so if you do that and you have present FAT-D, ATP, CoA, and oleic acid, you see conversion into oleoyl CoA. But this is limited by the amount of CoA in this particular case. So if you add more CoA, you also get more conversion. And so in the end, of course, this CoA needs to be recycled, but this is something I will get into later on. Now, not shown on the slide, but this is promiscuous enzyme, so you can feed all kinds of fatty acids, saturated, unsaturated, short chain, long chain, doesn't really matter. Can be fed and introduced into the pathway. So FAT-D is really a versatile enzyme in this pathway for synthetic cell. Then the next step is Pils B, and Pils B is a very particular enzyme. I think its structure was solved last year. It's a peripheral membrane protein which has a couple of amphipathic helices by which it can interact with the membrane, and this membrane interaction is essential for the activity of this enzyme. And that's also shown in this slide. Here we have purified PLSB, and we test the PLSB in an enzyme activity measurement. So when there's no membranes present, and we feed with oleoyl-CoA, we have glycerol 3-phosphate, and we have PLSB enzyme, there's no activity. You need to have a membrane. And the membrane composition, it's actually shown here, you need to have a membrane. The actual membrane composition is not extremely important, but it needs to be a kind of platform for this enzyme to bind. So it doesn't have much of a specific activity for particular phospholipids, but it needs to be a kind of scaffold. And this means that by using this enzyme PLSB and building a synthetic cell, we cannot build a synthetic cell by the novo synthesis of membranes. We need to have a membrane present in order to build a membrane. So there's a little bit of chicken and the egg, of course, but in this case, we work with pre-existing membranes, which are then further grown. Then the next enzyme is PLC, which we can add to this process. And then PLC converts LPA into PA, and then you have a real phospholipid. PA is the most simple phospholipid we can have in our system. And the advantage of having this pathway is not only that we build the phospholipid, but we also release the CoA in 2 steps, which can then be recycled via FAT-D into acyl-CoA. So you recycle, this pathway is then self-sustainable, where the conversion is determined by the amount of fatty acid we add, catalytic amounts of CoA, this is actually shown in this slide, but I don't want to go into the detail of that slide, and ATP. Of course, ATP is converted in AMP, and AMP has to be recycled. It's something I will address later on. But this is the most simple pathway to build phospholipids, and this leads to PA, which is normally in membranes a very minor component. So this phospholipid synthesis can be done on large scale. We have multiple enzyme turnovers in the assays we do. So we make chemical amounts of phospholipids. And this pathway is also compatible with any fatty acids you feed. I mean, it also showed this on the slide, but that's in the paper, which was on one of the first slides. Now, but we need a membrane to grow a membrane. So then the question is, do we really grow? Oh no, sorry, before I go into that, then the last part is the synthesis of PE and of PG. There we need a set of additional membrane proteins, including CDSA, which makes CDP-DAG from PA. And well, don't want to go into the details of this, but the reconstitution is such that you can, if you only include this part of the pathway, you only make PG. If you only include this part of the pathway, you only make PE. And when you, that's shown over here, PG and PE, if you mix these enzymes at different ratios, you eventually can get to a phospholipid ratio, which mimics more or less E. coli, which is roughly 30% PG and 70% PE. Okay, so by tuning the amount of reconstituted proteins, we can mimic E. coli membrane. But of course, this requires an additional feed with serine to make phosphatidyl serine, which is converted into PE by decarboxylation. All right, so do these membranes really grow? We use 2 different methodologies to look at the growth. One methodology is by using a self-quenching assay. It's an assay, it's called R18 assay, which is used a lot in membrane fusion fields. So this R18 is a very lipophilic rhodamine, which we can add to the membranes, and it self-inserts into these membranes, and by a certain concentration it will show self-quenching because the concentration is too high. If then the concentration is diluted, for instance by adding Triton, then you dilute the fluorescent probe, then you get relief of self-quenching and you see a fluorescent signal. This is the control with Triton. If we add fatty acids to our system, as we do in growing these membranes, you already get an expansion of the membrane because the fatty acids partition into the membrane. That's shown over here. So there's already growth, could say swelling of the membrane. And then by enzymatic activity, there's conversion to phospholipids. You do not get further growth because all these single fatty acids are converted into phospholipids. But at any rate, this assay shows that we have true growth also to the extent we expect in our feed. And so now the growth is only limited by the amount of fatty acid and other precursors we add. You can also look with electron microscopy at these vesicles. And what you see is that depending on the amount of fatty acid added, the morphology of the vesicles changes, they grow. But eventually, if you add too much, they also will fuse. So I think for growing in the synthetic cell, we need to be on the low end, preferentially continuous feed with fatty acids. We need to feed the fatty acids to a synthetic cell to grow. We cannot add too much because you will inhibit enzyme function and you cause fusion, as you can see over here. Further, another thing which is important is if the system is unbalanced and you make too much phosphatidyl acid, it forms complexes with magnesium, and these complexes precipitate. So what's really important in this synthetic pathway is that is formation of CDP-DAG, which is formed by CDSA, which is polytopic membrane protein, the most fragile protein in this pathway, and it's a rate limiting step. So that needs to be optimized not to get accumulation of PA. Okay, so in principle, we can grow membranes from the outside by reconstituting an entire phospholipid biosynthesis pathway. And the next step is to couple this to other modules for membrane growth and preferentially also to growth from the inside. Because in the end, the synthetic cell has to grow from the inside, would also have to grow from a DNA template. So we joined with Bert Polman, who's also in our institute. This work by Marta Exkaat and Jelma Koenradi and Illinois Balloni. So the first element is if you grow from within, you have to look at recycling of precursors. Because growing from within, accumulation of precursors and products, yeah? So substrates are converted into products and byproducts. These need to be recycled because often they inhibit progression in the enzymatic pathway. And there are a couple of molecules that need to be recycled. I already mentioned CoA, but CoA is recycled in the first step of this pathway, formation of PA. Then CoA is released. Then we get CO2 production and decarboxylation of phosphatidylserine. CO2 is also not a problem because it's exported by diffusion. But the key thing is, of course, nucleotide homeostasis, because we need ATP, we need CTP, and we also need glycerol 3-phosphate. So glycerol 3-phosphate can be done by different means. One can import glycerol 3-phosphate from the outside by including a transporter, but there's also a possibility to form glycerol 3-phosphate from ATP and glycerol. And then there should also be degradation of pyrophosphate, which accumulates at this step of CDSA. So in order to get a sustainable growth, you need to recycle all these components, which adds to the complexity, of course, of our pathway. Now, let me first focus on AMP and CMP, in particular CMP. Now, CMP is an end product of the polar head group attachment. CTP goes into this pathway and eventually CMP is released. And CMP has to be reconverted into CTP. And one way of doing that is using a cascade of kinases. The CMP kinase, which is a specific enzyme for CMP, and a nucleotide dikinase, which is a more promiscuous enzyme, can take any diphosphate nucleotide. So we use CMK, to convert CMP at the expense of ATP into CDP. And then the nucleotide dikinase can convert the CDP at the expense of another ATP into CTP. And then two ADP are released, but then they can also be reconverted by another pathway I get to in a moment. And that's shown over here. So here we have our synthesis reaction. Now we start from PA. We have CDSA, and we have, in this case, the phosphatidyl glycerol synthesis in this system. CMP is released. So this pathway normally works. You have oleic acid. This is a reconstitution. So we have oleic acid. We feed the proteoliposomes with oleic acid, and then we see conversion into DOPG, PG, provided that CTP is present and glycerol 3-phosphate is present, because that's used as a precursor for the polar head group. If we then feed with CMP instead of CTP, then the reaction doesn't take place. Then we have to include these two enzymes. So if we include those two enzymes, we can convert CMP into CTP, and CTP is then used by the CDSA to make the CDP DAG, and this is then eventually converted into PG. So in principle, this cascade works, and if you replace the CMK for AMK, then you can also use it to recycle AMP. The second recycling, the second process requiring ATP is glycerol 3-phosphate formation. So we can feed glycerol to the GLPK, which is the glycerol kinase, that forms glycerol 3-phosphate. And so, So this enzyme was also purified, and if you then provide it with glycerol and ATP in this system where the CMP is driving eventually the conversion, we don't need to add glycerol 3-phosphate, but we can derive the glycerol 3-phosphate from the GLPK reaction. Okay, now we want to run those reactions from within a large unilamellar vesicle. Now, I don't want to put too much time into this pathway because this was further developed by Bert Polman in our institute. He uses the arginine deaminase pathway to generate ATP by substrate level phosphorylation, you could say. It's a very simple pathway. There's a transporter, RD, which shuttles in arginine, and the end product of this pathway is ornithine, which is released by the same transporter, so we have a one-to-one exchange. Then there are a couple of enzymes that eventually lead to the formation of ATP at the expense of ADP. So you can make ATP inside these vesicles. Now, then we can look at glycerol. Now, there are, of course, glycerol transporters. E. coli has a glycerol transporter. But the question is really whether we need a glycerol transporter here, because glycerol is very membrane permeable. And that's shown over here in this light scattering experiment. These are large unilamellar vesicles, which are dropped into a high concentration of glycerol or potassium chloride. Now, they are dropped into potassium chloride, they shrink, and because potassium chloride cannot go into these vesicles, so the water is released by the liposomes, so they shrink. Whereas with glycerol, they initially shrink, but then they recover, and they recover because glycerol can permeate the membrane at a high rate, at least at rates which are by far surpass any catalytic activity we so far have been reconstituting. So we don't need glycerol transporter, but of course we need glycerol phosphate kinase, the enzyme. It's a multi-subunit enzyme, homo tetramer it is. Now, if we then generate ATP by the ARC-D system, then we can drive the formation of glycerol 3-phosphate in this system. And this is indirectly shown in this picture. Here, a fluorescent ATP-ADP indicator has been trapped into the vesicles. And if you don't have the GLPK in this system, so you don't make glycerol 3-phosphate, then the ARC system makes ATP over extended periods of time. These are hours. If you include glycerol and GLPK, then the GLPK consumes the ATP, and this is seen as a lower level of ATP generation in this system. Then we can put all these things together, includes part of the phospholipid biosynthesis pathway, but we start with PA, and PA is co-reconstituted into this liposome with a specific acyl chain composition so that we can easily monitor it by mass spectrometry. So we co-reconstitute ARC-D with CDSA and PGSA and PGPA, so to make PG from PA, right? And the ATP and the CTP, it's not shown on this slide, they come, so the ATP comes from the arginine pathway and the CTP comes from the regenerating system, which I explained in a moment, just before. So this system then works. You have PA. If you don't add arginine, you don't generate ATP, so then this pathway will not work. So you don't get production of PG. If you add arginine, the deaminase pathway makes ATP. The ATP is also converted into CTP, and the PA can then be converted into PG. I forgot to say we also have, of course, the glycerol kinase, so we can also make glycerol 3-phosphate, which is also essential for this process, and that's shown over here. So if we don't have the glycerol phosphate kinase included in this system, this pathway only runs up to CDP-DAG, because CDP-DAG cannot be further converted. If we include the GLPK, then we see the production of PG as we would expect. Now, of course, this is not bulk synthesis. This is tracer conversion in the membrane. So what we notice in this system, and it's not perfectly 50%, but we notice that our conversions are never complete. So if you look at the kinetics, we see a fast conversion stage, and this then levels off and it becomes extremely slow, and roughly 50% by approximation of the PA is converted into PG. And in this system, the conversion has to take place on the inner leaflet of the membrane, because that's where the ATP is. Inside, in the lumen of these vesicles on the inside, it's the ATP, so conversion can only take place on the inside. So what we think is that a major share of the PA, about half, is inaccessible to the system and likely requires some sort of flip base to become accessible. So one of our next stages is also to include all those kind of enzymes in these flipases into the system, adding again to the complexity of the membrane system. Okay, so this gives me a summary for this first part. So we can make bulk production of phosphatidylethylamine and phosphatidylglycerol by using purified membrane proteins. So in our systems, we can have a full conversion of the fatty acid feed as long as you don't add too much, otherwise the reactions are inhibited. We have CoA recycling, which is inherent to the pathway. There's a tunable acyl chain composition. I did not show that data, but that's you can add any acyl chain to the system. And there's also a tunable PE-PG ratio, mimicking E. coli membrane. Now, conclusion from the work was that we need pre-existing membranes to synthesize a membrane. This is, of course, not surprising because we use membrane proteins from E. coli and they need a membrane to be active. Now, we also observed that the extent of membrane expansion is just limited by the substrate availability. If we do it from the outside, we call this membrane farming. We can do ATP-driven glycerol 3-phosphate synthesis in the system, and also CTP and ATP regeneration. Didn't show all the data. However, although one can do phospholipid conversion from within the lumen of the vesicles, this is not yet bulk conversion. This would require much more. This would require that the fatty acids are also transported into the vesicles, and yeah, you would need a more complicated system. I will say a couple of things about that later on, how to approach that. Okay, now one of the other hobbies in my laboratory is to examine protein translocation. And so we use this, let's say, small synthetic cell to synthesize the phospholipids needed for protein translocation. So we used it to address certain questions in the protein translocation field. And in order to show you the data, I have to say a couple of things, or at least introduce you to this system. This is again a multi-component system. So E. coli and other bacteria, actually every living organism, they synthesize proteins on the ribosomes, of course, and proteins destined to be secreted are synthesized with the signal sequence. And in E. coli, this is a so-called post-translational process where the protein that needs to be translocated is synthesized to its full length, stabilized by a chaperone, and then targeted to the translocase, which is a large multi-component membrane protein complex. Now that translocase for protein secretion consists of a molecular motor, Sec-A. It uses the energy of ATP hydrolysis to drive proteins in a stepwise fashion across the membrane, and it does so at a pore complex which is termed Sec-YEG. I will not discuss the other proteins. The other pathway, and this is of course also important for a synthetic cell, is a co-translational pathway. This co-translational pathway is used for membrane protein insertion. So newly synthesized membrane proteins, again synthesized on the ribosome. Once the first TM domain emerges from the ribosome, it's bound by a signal recognition particle, which is a riboenzyme. Then this ternary complex of a ribosome with a nascent chain, so incomplete chain, and SRP is targeted to a membrane-bound factor, a GTPase, FTSY, which is reminiscent of signal recognition particle receptor at the membrane. And this FTSY targets this complex to the translocon, where the SRP and the FTSY are released at the expense of GTP, and then chain elongation on the ribosome is then followed by membrane protein integration via the translocon. Okay, so this is the system. I mean, the structure of that system has been, at least of the pore, has been resolved many years ago by the group of Tom Rappaport. And what's shown over here is a view of the translocon from the cytosolic face of the membrane. So you look from inside to the outside. Then you see the large Sec-Y protein, which is an internal duplication of 5 TM domains, which are connected through a flexible hinge. And this system can open on this flexible hinge. And then it actually opens more or less like a clamshell. If you would look at it like this, it can open like a clamshell on the hinge. And if it opens up like a clamshell, there's a plug domain which occupies the pore. It will fall out. And this whole structure is embraced by another membrane protein, SEC-E, which stabilizes this structure. So this is the basic structure. And if we look at the mechanism, this is a collection of structures from many different labs and an interpretation. So we have a closed channel. Now we look at from it as a cartoon from the side. We have the clamp of SEC-E, which embraces this structure. So we have this plug. I didn't mention this hydrophobic ring, which is not entirely in the middle of the membrane, but it's a kind of gasket. It seals off the channel. So the initial stages, the ribosome or SEC-A, they crack open this lateral gate. So it's partly cracked open. Then this ring, this hydrophobic gasket ring, expands. The protein will enter, and then it depends on what the modus of operation is. If it is membrane protein insertion, the lateral gate opens further, but the plug stays where it is. So the aqueous channel is actually blocked by the plug. And the idea is that membrane proteins slide into the membrane on this surface of the lateral gate with the lipid membrane. If it's protein translocation, this is done by SEC-A, the motor protein. SEC-A opens this channel much wider open, and the plug falls out, and then we get this kind of opening of the lateral gate, and the signal sequence etches into this lateral gate, and the remainder of the protein is then translocated through this pore. Now, something which has intrigued us already for many years is that there's a very strict lipid dependence of the SEC translocase, and this is for acidic phospholipids, which in E. coli is mostly PG or cardiolipin. Now, these acidic phospholipids, they are known to induce helicity in signal sequences, and this is important for translocation, so this is known. It's also known that the motor binds to acidic phospholipids on its own, and with its N-terminus, which is amphipatic helix, penetrates the membrane. And this then activates this SEC-A protein for high affinity binding to the pore, to the SEC-YEG system, and also activates it for ATPase activity. Now we also know that SEC-YEG, the channel, binds acidic phospholipids at its annulus. This has been done by isolating the SEC-YEG channel using block polymers, which capture a small piece of membrane with SEC-YEG in it. And then this is enriched in acidic phospholipids compared to the general phospholipid composition of E. coli. We also know that in vitro and in vivo, there's a significant lipid-bound pool. So we think that there's a diffusible lipid-bound SEC-A as an intermediate in the catalytic cycle. But this is not fully understood. So we tried to use our synthetic cell system to address some of the questions and also to separate more or less the lipid dependency of SEC-YEG from the lipid dependency of SEC-A. And one way of doing that was to reconstitute the channel SEC-YEG into a phospholipid composition that does not support protein translocation. And that's phosphatidylethanolamine and phosphatidylcholine. There the system is inactive. This is an assay where we assay translocation. I'll explain in a minute. But that system is inactive. And we co-reconstituted the channel with the phospholipid biosynthesis genes, proteins. Right, so it's co-reconstituted. So there are quite a few proteins there. And then eventually we add SEC-A because that's a soluble protein. And we add a precursor protein, which in this case is precursor for OMP-A and ATP. And then if the system is active, then the OMP-A is translocated by SEC-A and SEC-YAG into the vesicles. And this is then assayed by a protease resistance assay. So we add protease on the outside that eats away all the non-translocated protein, but has no access to the translocated protein, which is fluorescently labeled. So as shown here, when the SEC translocon is reconstituted in PEPC, it's inactive. When it's reconstituted into a liposome, which also has phosphatidylglycerol, about 30%, it's fully active in translocation. So it needs PG. So we took those liposomes that do not support activity and then synthesized PG with our phospholipid biosynthesis pathway. And that's shown over here. We can synthesize PG to a certain extent in this system. Start off with oleic acid, and then in the end get PG. About 25% of the total phospholipid is PG in this membrane. And then what actually happens is that we revive translocation. So in PEPC and oleic acid, where there's no conversion, the system is still inactive. But if we make PG biosynthetic cell-like mimic, we reactivate the system. And this helped us to draw a certain conclusion. I cannot show all that data on the SEC system. I collaborated with Sivajan Mark in our institute. Cesar Lopez, a postdoc in this group, did some coarse-grain MD simulations on this system, the SEC-A, SEC-YAG complex in a phospholipid bilayer, reconstituted in a phospholipid bilayer. And they examined the frequency of occupancy of certain sites on SEC-YAG with PG. And to make a long story short, I had 3 sites. One of these sites is a site where the N-terminus of SEC-A is. This is the N-terminus of SEC-A. Here we look from the side solid phase again. This is a site which becomes enriched with acidic phospholipids. This is exactly where this amphihepatic helix is. And so we think that SEC-Y binds acidic phospholipids at this region, and this helps SEC-A to be activated through the action of its amphihepatic helix. Okay, this brings me to the model which was derived from those experiments and many other experiments. So SEC-A is a soluble protein. It can bind to the membrane to acidic phospholipids. When it binds to acidic phospholipids, it's primed for high affinity binding to SEC-YAG, and it's also primed for ADP-ATP exchange. So ATP binds to SEC-A, and then SEC-A undergoes a conformational change, which is fully understood, which drives segments of a polypeptide across the membrane. And this cycle is repeated many times to translocate the entire protein across the membrane. So this is where at least our synthetic cell mimic helped us to address specific questions in our research. So the last part is about reconstitution of the SEC translocon in the giant unilamellar vesicles. Because we think that building a synthetic cell probably needs to be done from a large vesicle that can efficiently trap the DNA template and all the enzymes needed for conversion, have a large membrane surface. And we also think that many of the proteins we need in our synthetic cell are membrane proteins. So we think that we need the SEC translocon to have efficient maturation, biogenesis of those membrane proteins. And the principal idea is that if you take a liposome which has some SEC-YAG, you can ignite insertion of membrane proteins, including SEC-YAG itself. Right, so this is the idea. Many of the proteins we work on are membrane proteins. Many of the phospholipid biosynthesis proteins, they are membrane proteins and they require SEC-YAG for insertion. So let's say the future synthetic cell should be a cell that's programmed by an genome, simple genome. And that should lead to the synthesis via an in vitro transcription translation system of newly synthesized membrane proteins that need to insert into the membrane via SEC-YAG. So this can either, this in vitro transcription translation system can either be a lysate or a pure system. Pure system is nicer than the lysate, but the lysate are usually of higher fidelity. Now, keep in mind that in vivo membrane protein insertion is not a spontaneous event, but it's mediated by the translocon. That's different in a liposome. If you have an empty liposome, there's always a tendency of membrane proteins, when they're synthesized on the ribosome, to insert spontaneously. So this spontaneous insertion is really a phenomenon that does occur, but it's very typical for in vitro systems where you have a large membrane surface. So the question in the future will really be, will the spontaneous insertion be sufficient, or do we really need the translocon? So we think that a minimal synthetic shell should be capable of efficient membrane protein insertion. Because if you don't have that, many of those membrane proteins synthesized on ribosomes, they will aggregate and will cause all kinds of trouble in the system. So to reconstitute SEC-YAG into giant unilamellar vesicles is not a trivial thing. So we actually used a method which was developed by others, emulsion-assisted method for guff formation. The method is a little bit adopted. I'll explain in a moment how. So we make small unilamellar vesicles, or luvids, with the correct phospholipid composition. These are then detergents destabilized with Triton, and to that SEC-YAG, membrane protein is added. And then through biobead reconstitution, we make small luvids or surfs with SEC-YAG. Now, this system is then mixed with a fluorinated oil, and then you get a kind of emulsion with water droplets in which the surfs are enclosed. And in this system, Krytox is added. The Krytox is a very anionic molecule, and that captures the magnesium and causes these vesicles to fuse. So then you get a fused vesicle. Then eventually you have to release these vesicles from this emulsion. This is done by the addition of perfluor-1-octanol, which causes a phase change. And then you get the emulsion separated from the aqueous solution, and the aqueous solution contains the guffs. And now, of course, the question is whether the SEC system survives this process. But first, let me say what we modified to the method. The original method is a mixture of reconstituted protein and empty liposomes. We use only reconstituted liposomes. And this is done to get sufficient amount of material, but the drawback is immediately that guff formation is far less efficient when you do it like that. If you add empty liposomes, it's very efficient, but once you use liposomes which have a reconstituted protein or proteins in it, it becomes far less efficient. And that can be seen in the yield. Now, this is a fluorescent image of our guffs, where the lipid membrane was stained with a fluorescently labeled PE, and SEC-YAG was labeled with a fluorophore, an ATO, also an ATO fluorophore. And when you then illuminate at the correct fluorescence wavelength, you can see that the guffs have SEC-YAG in it. Okay. So these guffs are quite large. They are roughly in the order of 30 microns, and they're also very stable. But one of the problems is that the guffs are still contaminated by small unilamellar vesicles, which contain SEC-Y. So in order to get rid of those small unilamellar vesicles, we included in the GUF formation high concentration of sucrose. Now, in this fusion process, this is a very leaky process. The sucrose is trapped in the lumen of the GUFs, but is not trapped in the soffs, which do not participate in the fusion event. And then by density centrifugation in another solution, you can spin down the GUFs, and that's shown over here. So we have the GUFs made, we spin them down, so the GUFs are in the pellet, or SAC GUFs, which have SAC YEG and the pellet, and not so much in the supernatant. If we use control SUVs, the FUVs are not pelleted under these conditions, but they are in the supernatant, and that same holds for the SAC SUFs. So we collect these, we concentrate as pellets, and then test them for protein translocation using a similar assay as I depicted before. So we have SAC YEG in the GUFs. We add SAC A, which is soluble protein, we add ATP, and we add a precursor protein OMP, which is fluorescently labeled, and then test translocation by proteinase K resistance assay. And that's shown over here. These are the SOFs, so the SOFs are very active. These guys here, they're very active. If we don't include SAC YEG and we collect the SOFs, we collect the GUFs, we have very low activity, only some aggregation binding. And if we collect the GUFs, they are very active. And this tells us that the system survived this, more or less critical treatment of GUF formation. That can also be seen in fluorescence imaging. So we can translocate the fluorescent OMP into the GUFs and then visualize it by fluorescence imaging. Although the picture might not be so clear, the analysis is much more clear. So if we don't add SAC A, we only get, these are the blue ones, so purple, what is it, purple. We do not get much translocation or binding because it's difficult to discriminate between binding and translocation here. If we add SAC A and we add ATP, then eventually in time, a greater amount of the OMP ends in the lumen of those vesicles. So in principle, the system is active. So this brings me now to the outlook and the challenges for the future. So I think one of the big challenges will be to have continuous and bulk phospholipid biosynthesis from within liposomes. It's my opinion that this will be very difficult to do that by a reconstitution technique, which I have been talking about mostly. The reason being is that if you have peripheral membrane proteins, they don't like to be, what we see from practice, they don't like to be entrapped in the liposome. We see a lot of binding of peripheral membrane proteins to the lipid surface, and they resist internalization. So in the end, of course, high performance genome-driven phospholipid biosynthesis is needed in liposomes, either by an entrapped lysate or using a pure system. Of course, this is of course a formidable challenge still. I think there have been some examples where this pathway, also in our basic program, has been reconstituted from pure trapped in LUFs, but this only leads to very small amounts of phospholipid biosynthesis. In the end, bulk phospholipid is needed. So this is a big challenge for the future. Then we need to have functional integration of the phospholipid biosynthesis pathway with other modules. Energy generation, I showed an example. The first step to WISE. The SAC translocase, specifically for membrane protein integration, something we work on right now, and eventually, of course, cell division. Okay, this brings me to the end. Acknowledgements, people in my group, Marta Exekate and Sabrina Koch. Marta Exekate did a lot of the phospholipid work, together also with Niels de Kock. Sabrina did the work on the SAC translocon, together with Martin, and Jelma did the recycling system. And Tim van der Akker and Max Nel did the GUFs work together with Ludo Skoepmakers of the Radboud University in Nijmegen in collaboration with Willem Huck. So we collaborate with Bert Polman, the PhD student who now has left, Eleonora Baroni, and with the molecular dynamics group, Cesar Lopez and Sivijan Mark. And I thank you for your attention, and I'm looking forward to the questions.
Thank you. Thank you very much. That was a fantastic talk. There's quite a bit of questions in chat. Can you see chat?
Chat, I have to open them. Can I stop sharing, right? And
Yeah.
I can see
Yeah.
the chat. I didn't see any hands, so earlier.
Yeah, people put questions in chat.
Yeah, that's fine. Okay. Okay, start. Already very early. How would you propose to deliver redox potential to synthetic cells, building membrane gradients or some other more direct ways? Well, membrane gradients is a little bit less of an issue. You can, of course, include the 1FO ATPase to pump protons across. Also, the arginine deaminase pathway makes membrane osmotic gradient. It actually consumes protons. And so it actually makes both an electrical potential and a pH gradient. The question will be if that's sufficient. About the redox potential, that's still quite something. There are systems to do redox shuttling. That's also done in the BASIC program. But we are still looking for the proteins we need that need this redox shuttling. Let's put it like that. A synthetic cell is still very simple. And every month, of course, another protein needs to be added to this system. So it will get quite complicated. But so far, there's not been a requirement for redox. But I think eventually it will come up, I suppose. I think here the big challenge is to keep a sealed system, because what I did not tell is if you take those GUFs, the GUFs we use, they are in principle sealed. But when you start to include in vitro transcription translation systems, these membranes become very leaky. And that's, I think, one of the real big challenges. If they are leaky, then you cannot maintain gradients. Oh, yeah, does that address the question of Andreas? Yeah.
He can speak up if you didn't, and we can move on to the next one.
Exactly. Okay, could you please elaborate on the concern about using too much fatty acid because it will inhibit enzymes? What protein enzymes do you see being inhibited by the fatty acids? Now, actually, the pathway is inhibited by too much fatty acids. We have not really studied the exact reasons why this is. But what happens if you add a lot of fatty acids, the membranes become very leaky. And I think you start to solubilize the proteins again. And I think the key candidates which would be sensitive to this is PLC and PLC, but we have not really studied that. We have studied more the permeation of fatty acids, and permeation of fatty acids at low concentration could be a major bottleneck. So I think there we need a transporter also to bring fatty acids in to the system in an efficient way. So I think in a synthetic cell, we really need to feed with low concentration of fatty acids and prevent that we use these millimolar concentrations that eventually become inhibiting. So this synthetic cell of the future will be a very spoiled synthetic cell because it will get all the goodies that it needs. Yeah? So it will also be very crippled, of course, but it will be really spoiled with all food sources, because that's the most easy way to get to this synthetic cell. Okay, then we have, in order to assemble the proteins on membranes, they were taken with parts of E. coli membrane, or they were purified, right? And they were purified, they were overexpressed and purified with this text. Also, could be possible to use the same machinery to make a double membrane like gram negatives. Yeah, that's that's a tough one. I think I think there are now some examples where one can make a mimic of a double membrane, but this is not with a liposome. This is with sheets, with nanodiscs. To do that with liposomes, I think it's not too difficult to trap a liposome into a liposome, so to have two membranes, but then to reconstitute, I don't know if that's possible to reconstitute a complete system that spans both membranes. I think that's still quite a challenge, but I'm sure people are working on it. I guess it's not impossible, but it's not something we have done. What's the kinetics of the Sec translocon? Well, okay. Well, the kinetics of the translocon principle not so bad. I think we can reach something like 1 to 10% what's in vitro. I think what will be important here for the Sec system and the synthetic cell will be the kinetics of membrane protein insertion. And so we're doing experiments on that, and it seems that insertion itself is not limiting, but the translation is limiting. So I think most of the challenges, that's my personal opinion, will lie in the in vitro transcription translation. To get that really in pair with the sufficient rates. That's what I think will be one of the big challenges, including trapping this system, in vitro translation, in sufficient concentration in a liposome. Those are the real big, tough.
That will be tough.
Issues.
I'm really looking forward to that.
Yeah, I always tell my students, if you would have a spoon and you put that spoon straight into the cytosol of E. coli, it will stay upright. The viscosity is enormously high. But if you do that in an in vitro transcription translation system, it will just collapse.
Yeah.
And so we're far off from the concentrations of protein that can be reached inside of cell. So that will be really, I think that will be the biggest. That's for the new generation. That's that challenge.
I'm really looking forward to that. Thank you so much for answering all the questions. I think we should wrap up because we're way past time. I want to say thank you.
Dinner.
Thank you so much. Thank you so much, Arnold. And thank you everyone for a great discussion.
And you guys all, good luck tomorrow with the elections.
Oh boy.
Good luck. I will.
Fingers crossed.
From a distance. Okay. Take care. Okay.
All right. Have a great rest of your day. Thank you so much.
Bye-bye. Thank you.
Bye.